In a revision stream today, and you might be like, Joseph, why? What is the actual point? Not only are you 3 days away from school holiday for a week, but after that, what is there to be revising for? Well, we actually do have mock exams in, I believe it is... If it's the 13th today, we have just under 3 weeks before mock exams. And while that might not sound like too much of a big deal, it certainly is. It's quite a short amount of time, especially if this full, if what we're going to be revising for is the whole of paper 1. So, without that, I'm going to basically be just going onto the AQA website. I'll pick a past paper and we'll try and do as much of it as possible. We'll try and just do it. So I am going to be streaming Cuphead in about half an hour. So while I might not have too long to do this, I don't care. I'm still going to give it a go. So we go Science AS, and I want to be doing the AS exam. So that's the question papers and mark schemes. Component, paper 1. Standard. Qualification, AS. Okay, so we got one from... we'll do the one from 2020, because 2020 was a fucked up time. Let's see how fucked up this paper gets. So that's a 2020 paper. Again, I cannot guarantee that I'll know any of this. Oh, and great, the first question is one that I literally know. I've revised this question about 50 times now. It's burnt into my head. It's burnt into my soul. Okay. This question is... And I completely forgot alpha and beta glucose. I think beta is the one where OH is on the top, I believe. I forgot. I've literally forgotten this. I'm trying to balance my computer so it doesn't fall. My apologies, guys. Okay. It shows ATP, alpha glucose, beta glucose, and protein. Just a protein. And we need to find out which one is a monomer in an enzyme's active site. Well, that would be the amino acid. Sorry, not a protein. That was so silly of me. It's an amino acid. You have the carbon, the amine group, the functional R group, or variable R group, and the carboxyl group. So that's B. Is a monomer in cellulose, that would be D, that would be beta-glucose. Is produced during photosynthesis and respiration, that would be ATP, energy, which is C. And forms a polymer that gives a positive result with a Bayou-Ray test. Now, Bayou-Ray test is the protein test, purple protein. If it forms a positive result, that's going to be B, the amino acid. Can we just make sure we got that right before we continue? B, D, C, B. Nice. Nice and easy. Okay. I feel like I've done oh my god, bro! This is we've done all of these questions. Holy shit. This is the Raffinos question. So you got C6, H12, O6, I believe. Yep. And that's just repeated for each monomer. Each monosaccharide, sorry. Not each monomer. Well, technically it's a monomer. And what we're saying, and I'm going to try and explain this so that you guys actually know what it means. So what it's saying is that this raffinose is all of these joined together. Now, if they're joined together, that's via a condensation reaction. That's the only way that they can be joined together to form a polymer, yeah, a polysaccharide. If they're joined together via a condensation reaction uses 2 hydrogen, 1 oxygen. So in order for this to be correct, I guess it's maybe best if you try and draw it. I'm not that good at drawing, I'll be honest with you. Basically, if they're joined up like this, you are going to need to subtract from the formula 2 H2Os. If it's joined together in a chain of 3, then the condensation reaction is occurring there and there. So that is 2 H2Os. So what we do is we add them all up. So that's C18, H36, O18. Because it's all of them combined. But we subtract 4 hydrogen. So that's 32. And 2 oxygen. So 18, 32, 16. And that should be the correct answer. 18, 32, 16. A biochemical test for reducing sugar produces a negative result. So reducing sugar, so that would be, if it's non-reducing. It's asking us to find the non-reducing. Non-reducing is that extra step where you have to hydrolyze the glycosidic bonds. Hydrolyze, neutralize, and then obviously add your Benedict. And remember, if it ever asks you to describe a test of any kind, a food test, whatever, always, and I mean always, you have to give a positive result. So a positive result in this case would be brick red precipitate. Hold on. Would it be precipitate? Or would it just be... No, it would be precipitate. Okay. So, in order for... This is 3 marks, and you need to talk about heating. So a good thing you could say is... adds acid... Heat with acid. There you go. Then add an alkaline, or neutralize. Add your Benedict solution. A positive result will be the formation of a red precipitate. Let's just check if we got that right. Heat with acid and neutralize, so that's 1 mark. Heat with Benedict. Okay, so you should have. You should have also said heat with Benedict and a red precipitate color. Nice. It says accept boil, which we again just put heating. So heat with acid, neutralize it. Heat with Benedict solution, red precipitate. That is a nice and easy 3 marks. Explain the arrangement of phospholipids in a cell surface membrane. Okay. I have not done this question, so I'm gonna 2 marks. Seriously, 2 marks here. And this is the formation. What it is holy shit, how have I forgotten this? Phospholipid bilayer, and it literally says phospholipids. Okay, so it's 2 layers of phospholipids. The hydrophobic tails face inwards. The hydrophilic heads face outwards into the actual cell, I guess. That's a tough one to describe. I guess you want to talk about hydrophilic/phobic. I don't know what else you would talk about there. Just the fact that the tail was kind of like in the fatty acid. Oh, I guess fatty acid chains and the glycerol heads. I don't know what else you would talk about there. That's kind of a weird question, actually. 2 marks, and not that bad. Oh, seriously? Oh, you're literally taking the piss. You guys want to know what the answer was? Boiler. I brought 2 letters. You know what? That is a really scummy question, though. Because you just assume. Okay, it says explain the arrangement of phospholipids. You already think phospholipid bilayer. You don't even need to say it. So I didn't say it. But you did, okay. That is a cheeky question right there. Literally saying, describe a phospholipid bilayer, and one of the marks is saying phospholipid bilayer. That is a cheeky little question, which I would have gotten 1 mark on. So you would have only gotten a mark for talking about, you could have also drawn a diagram and wrote the word bilayer. So my thing here, saying 2 layers, I could have potentially gotten a mark for that. Oh, interesting. Accept 2 marks for an annotated diagram of bilayer with water labelled on each side. That is a really interesting one. So if you could have literally gotten a mark for saying water, bilayer, and then maybe fatty acid, there, something like that. But okay, so what you really should have said is the structure of the phospholipids forms a bilayer because the hydrophilic glycerol molecules face towards, or I guess they tough one to explain. I guess you could say the fatty acid tails they are repelled from the water, the heads are attracted to the water. I guess that's that. So all you would have had to say is bilayer, that's attracted, that's repelled, the heads face the water, the tails face inwards. There you go. Tricky little question there, because you really could have maybe failed it. Describe how an ester bond is formed in a phospholipid molecule. Huh. Okay. I'm going to be completely honest. I kind of blanked there. It is... You have the glycerol... No. I'm thinking of a triglyceride. You got... Is this not what it is? So you have... So wouldn't you literally just say a condensation reaction joins together the fatty acid and the glycerol? Will that not be both marks? I'm confused. What else was it asking for? Condensation... Oh, wow. Okay, that was literally it. Condensation between glycerol and fatty acid. That was literally that. And you could have literally said maybe condensation between, obviously, the fatty acid. I'm just going to write F acid. Oh, wrong one, sorry. That's for glycerol. And your fatty acid. Nice and easy question, actually. Again, you see how these questions get you to overthink things. Because some of the questions are asking for the bare minimum. Like that one was asking for the bare minimum. Literally condensation between that and that. The other one was asking for quite a lot. Explain the property of water that helps to prevent temperature increase in a cell. Ooh. Prevents temperature increase. Wouldn't that be latent heat of vaporization? I knew that one. Specific heat capacity. So the buffers changes in temperature. Or in other words, a lot of energy is needed to change the temperature. That was nice and easy. Water has a specific... I'm just going to write water has a high specific heat capacity. Nice, literally That could get you 2 marks. That is obnoxious. Describe how a phagocyte destroys a pathogen present in the blood. Now, we have actually done this. And I don't remember. Holy shit, why don't I remember? This is literally the topic we've just started, and I don't remember. Describe how a phagocyte destroys a pathogen. Well, it's asking for destroying. Lysosome. Releases an enzyme. Shit. How do I not remember this? We literally did it last week. This is why we need more biology lessons, apparently. Great. Lysosomes. Okay. They release a specific enzyme, and I don't know what the enzyme's called. Lysozyme, that's it. So lysosomes release... Oh, they trap the bacteria or the pathogen in a vesicle. So it's engulfed by the phagocyte. The pathogen is then surrounded by a vesicle. Lysosome releases the enzyme lysozyme, which hydrolyzes the pathogen. That's going to be my answer for a 3-mark question. Let's see. Engulfs, forming vesicle, fuses. Oh, that's it. The vesicle fuses. I'm going to keep this up. So it's engulfed, a vesicle is formed around the pathogen, which fuses with the lysosome. The enzymes digest... Oh, no! No way! You wouldn't have gotten a mark for saying lysozyme? That is some bullshit. So you could have put enzyme, i.e. lysozyme. That is a nasty marking point. And you wouldn't have gotten a single mark for talking about hydrolysis. Oh, no, you would have. Enzymes digest / hydrolyze. So enzymes hydrolyze / digest. That is actually quite easy. The phagocyte engulfs the pathogen. A vesicle forms around the pathogen, which fuses with the lysosome. The lysosome then releases enzymes which digest and hydrolyze the pathogen. You wouldn't have gotten a single mark for talking about other stuff. Okay. Give 2 types of cell other than pathogens that stimulate an immune response. Oh. We don't know this. We have not done this. So I don't know. Would foreign cells, or is that too vague? Is that too ambiguous? Because I know foreign cells would trigger an immune response. What else? Non-pathogens. Like stem cells? I don't know. Let's find out. Huh. Okay, that was an obvious question, actually. Literally, you could have said cancer cells. Cells infected by virus, we don't really do much on that. Like, we haven't done much on that yet. Cells from other organisms. So I put foreign cells. That wouldn't have counted. You would have had to be more specific and say cells from other organisms, such as transplants. That's nice. That's nice. Figure 2 shows the structure of an antibody. Label figure 2 to show where an antigen-antibody complex forms. We have not done this. Skip. What is the role of a disulfide bridge in forming the quaternary structure? Disulfide bridges form between the cysteine amino acid. Would that maybe give me a mark? Let's find out. Oh no, they just join together 2 different polypeptides. I didn't know that. I'm going to write that down. disulfide join together polypeptide 1, polypeptide 2. I did not know that about disulfide bridges. That's really cool. Right, final one and then we're done. Eukaryotic cells produce and release proteins. Outline the role of organelles in the production, transport, and release for 4 marks. And it says don't include transcription and translation. Alright, let's think about this logically. If it's asking for 4 marks, I like to always multiply that by 1.5. So we're looking for 6 points here, max. The role of producing and releasing proteins. Well, we know that obviously in order for proteins, it's protein synthesis. Whereupon, I mean, it's asking no details, so I'm just going to put where RNA is used as a template to produce amino acid, to produce polypeptide chain. That's 1 point. Number 2, transport release. Okay, well we can say exocytosis. That's how it's the release of proteins. We could talk about the obviously vesicles in the Golgi. That's how that works. So same point actually. Same point there. Vesicles are used to transport the proteins. They are, I guess you could say they fuse with the cell membrane, releasing the proteins. Next. You could talk about the Golgi itself and how that forms proteins by combining it. It's checked and modified. You could probably include info about the rough endoplasmic reticulum and how that produces or synthesizes proteins. I don't know much else apart from that. So you know what? Let's check the mark scheme. We got rough endoplasmic reticulum is involved in protein synthesis. RNA is used as a template to produce the polypeptide chain. The Golgi body checks and modifies proteins by adding, I'm going to say, glycosidic chains. Then the vesicles from the Golgi body are used in exocytosis. I don't know how you would word it, but let's just check if we're on the right page. Yep, I was. DNA in nucleus is code. Okay, so right away, DNA, not RNA. Ribosomes produce protein. So again, 1 mark. Mitochondria produce ATP. Interesting. I forgot protein synthesis requires ATP via mitochondria. So mitochondria, 1 mark. Golgi apparatus, package and modify. Okay, not checked and modified. Package and modify. Carbohydrates are added that are produced. Carbohydrate added, so glycoproteins. Okay, that was interchangeable. The vesicles transport. Yeah, 1 mark. The vesicles fuse with the cell surface membrane. Okay, forget exocytosis. Cell surface membrane. Okay, so DNA is used to code for proteins. Proteins are synthesized by the rough endoplasmic reticulum. I guess, again, mitochondria produce ATP for protein synthesis. The Golgi apparatus is the thing that actually it packages and modifies. That's an important word. I'm going to circle that even. It packages and modifies the protein. Vesicles transport it, and the vesicles fuse with a cell surface membrane, which releases it outside of the cell. Honestly, I might have gotten all 4 marks, and I kind of waffled. So I would probably give myself 3 out of 4 for that, but considering, for the most part, we haven't talked about that in a while, I'm happy with that. This is a plant cell, and it's asking why a nucleus is not visible. Suggest, so it's not fact. Couldn't you just say the nucleus hasn't absorbed any of the stain? It's an electron microscope, hold on. I don't know. Oof. But it's a 1 mark questions like that really fuck you over. And I don't know. My best guess would be the stain hasn't been absorbed into the nucleus. Alternatively, a more logical guess would be that plant cell doesn't have a nucleus, but it's likely not going to be that, is it? Yeah, nucleus not stained. Okay. Name the organelles S and T. S is the cytoplasm. T is it's pointing at the chloroplast. So. Oh, hold on. That was a vacuole. Huh. Vacuoles are pretty damn big, apparently. Huh. I didn't know that. That's actually really deceptive, because on my screen it looks completely clear, but it's not. That's apparently the vacuole. That's cool. Give 1 advantage of viewing a biological specimen using a TEM than an SEM. Less risk of artifacts appearing, which would, I guess, ruin the image. Maybe. No! Oh, that's the wrong way around. I'm pretty sure scanning is the way. No? Oh, literally, come on, that was so obvious. High resolution. Higher. Calculate the number of cells in the upper surface of the leaf. That's a nasty question. 2 marks for math. Can't do those ones. Two solutions use to stain tissues. So, we can't do that one yet. Can't do that one. Describe how messenger RNA is produced from an exposed template. Do not include DNA helicase. Okay. Messenger RNA is produced. We've done this today. We cannot fail this. Okay. So the DNA template gets unwound. Okay. The DNA template is, the DNA strand is unwound. Free floating RNA bases are attracted to the, I guess, exposed bases. Number 3, this forms together messenger RNA because only a single strand of the DNA is used as a template. I'm going to write something like that. Let's see what the answer is just before I head off. Free RNA nucleotides from complementary base pairs. Okay, we got no marks. The free RNA, so free RNA, not bases, nucleotides. The free-floating RNA nucleotides form those base pairs. phosphodiester bonds form. And then that is because of RNA polymerase. So that's how you get your messenger RNA. Okay, so in summary, when the DNA is unwound by DNA polymerase, DNA helicase, it unwinds the DNA. The free-floating RNA nucleotides bind with the complementary base pairs, they bind with the complementary bases to form base pairs. The phosphodiester bonds then form because of RNA polymerase. Okay. Define the term exon. Exons are not removed, they are coding. They are coding bases, which means they are spliced together during, I guess, DNA splicing. I don't know, what's the definition? Tell me. They are a base sequence coding for the polypeptide sequence. Okay, so all it was asking for there was the definition of a gene. It's a sequence coding for a polypeptide sequence of amino acids. It's the base sequence coding. That is a tricky one, actually. Define the term exon. So exons are literally just the base sequence from which polypeptide chains are formed. Alright, it is almost past, so I'm heading off now. Decent revision. I'm happy with that. Water has a high heat capacity. Disulfide bridges join together polypeptide chains. We now know a little bit more about phagocytosis. DNA gets unwound. The free-floating RNA molecules form complementary base pairs. Phosphodiester bonds form because of RNA polymerase. We now know that exons are bases which form specific amino acid chains or polypeptide chains. We now know a bit more about protein synthesis. I'm happy with that. That's been fun. Goodbye.